Hi, everybody. I'm Archie, as Harini mentioned. I'm a third-year PhD student in biomedical engineering at Yale, and I'm going to present about our work in the co-profiling of the transcriptome using DBiT-seq and the proteome using the PhenoCycler Fusion system. So DBiT is a technology that was published in my lab. It was first used to profile the transcriptome, and it primarily works using a microfluidic based system. So we flow a series of barcodes through these PDMS microfluidic devices, and that is how we can barcode both the transcriptome. We can also profile the transcriptome and the proteome in spatial site-seq. And we've also profiled the epigenome in spatial ATAC-seq and spatial CAD tag, respectively. And we've also profiled the epigenome and the transcriptome as well. So it is a quick overview of the DBiT technology. So it's applicable to both FFPE and fresh frozen samples. So when you start with your tissue section on a glass slide, and then you go through the various preparation steps, so fixation, permeabilization, and then we flow in two sets of barcodes. So the first barcode, these barcodes will determine the position of the transcript. So these barcodes are flown orthogonally. So you end up with a grid, and you end up with a 2500 pixel grid, and each pixel contains either the transcript or the epigenome or the proteome that you're looking at. And the resolution of this technology depends on the width of the microfluidic device. So it can range from 10 microns to 50 microns. I'm not going to bore you with the details of the PhenoCycler Fusion system, but it's a multiplex immunofluorescence approach that utilizes the addition of fluorescently labeled DNA, using fluorescently labeled DNA probes that bind to conjugated DNA conjugated antibodies to visualize protein markers. So the premise of my talk here is how to combine our DBiT-seq approach with the PhenoCycler Fusion workflow. So in the DBiT workflow, in the very last step, after the barcoding process, we proceed to lyse the tissue, and then we can do downstream steps such as template switching, PCR. We build a library for sequencing. But if we want to combine our DBiT workflow with the PhenoCycler Fusion workflow, we have to make sure we still have our tissue section intact on the tissue slide. So we we tried different approaches to do this. The first approach we tried was a chemical approach. So we tried using sodium hydroxide and DMSO, and these are known agents for denaturing DNA. And we also tried an enzymatic approach using RNase-H, which can selectively degrade RNA in a DNA-RNA hybrid. So for sodium hydroxide, we tried different concentrations and different incubation times. The biggest pro here is that we get pretty decent gene and UMI count, but the main disadvantage of this approach is that the tissue morphology is disrupted, so you can't really use that for downstream multiplex immunofluorescence imaging. DMSO also works similarly to sodium hydroxide. It can also denature DNA. And we also tried different incubation times and different temperatures. But the issue with DMSO is that you get even lower gene and UMI count compared to using sodium hydroxide. And the tissue morphology is also disrupted, so you don't get really good multiplex immunofluorescence data. So we decided to try a more elegant approach, which is using RNase-H. So as I mentioned earlier, RNase-H can recognize a DNA-RNA heteroduplex, and it can specifically cleave the RNA section of that. So this image just shows how RNase-H works. So we tried different RNase-H enzymes. We tried different incubation times and concentrations, and we found that the tissue morphology was preserved. We also had very good gene and UMI count and very high quality multiplex immunofluorescence data that I'll show going forward. So this is just showing how the sodium hydroxide approach worked. So the first image on the on the left is the image of the tissue section before our barcoding process. So this is a mouse embryo E11, and the middle image is after barcoding. And the image on the right is after the cDNA collection. So we incubate the tissue with 0.05 molar sodium hydroxide for an hour, and then we collect I call it the elute. But what you see here is that you tend to have issues with tissue detachment. So the tissue detaches from the slide because it's a pretty harsh approach. So we switched to RNase-H. I've glossed over a lot of details. This took quite some time to do, almost a year to pivot towards this new approach. So initially, after our DBiT workflow, we would incubate the section on the slide with 10 units of RNase-H for 37 minutes, at 37 degrees for 60 minutes, and then we'll collect that, and then we'll do DAPI stain and store the slide at -80 for the downstream PhenoCycler Fusion workflow. So what we found was that this particular enzyme, we were getting pretty low count, so we decided to switch to a different RNase-H enzyme, which was thermostable. So we switched to the NEB version of the RNase-H enzyme, and this specific enzyme has higher activity at higher temperatures, and it can range, its optimal activity ranges from 37 to 95 degrees Celsius. And we also decided to pivot towards using FFPE samples because fresh frozen samples tend to be more delicate, but FFPE samples are very robust. And in the FFPE workflow, you subject the sample to antigen retrieval, which is at a pretty high temperature, and the section is still fine. So that was the main pivot. So we decided to use primarily FFPE samples. And we also increased the concentration of the enzyme from 10 units to 20 units and increased the incubation times as well. So this is some data from a mouse embryo. This was a fresh frozen mouse embryo. So this was a test or a proof of concept. So on the left is a test condition. So we use the RNase-H enzyme, and on the right is a lysis. So in this, so these are serial sections from the same block. So on the right, we lyse the tissue after the DBiT workflow. We didn't perform multiplex immunofluorescence staining on these slides though, but we see that even though the mean UMI and gene counts are much higher for the control, but as I'll show in the subsequent slide, we are still able to recapture the general anatomical structures of the mouse embryo. So this is an image of the mouse embryo, and then this is the spatial clustering. So we performed the clustering using Surat, and the clustering also tends to overlay pretty well with the spatial structures in the mouse embryo. And we found 8 unique clusters, and they're very specific. For example, we see clusters that are very specific to different structures of the brain. So we also compare that with the lysis, and we also see pretty similar spatial organization. So from this data set, we were pretty confident we can use this approach using the enzyme to get very good transcriptome data, because we will need the transcriptome data to combine with the proteome data to give us a better understanding of what is happening within the tissue. So as validation, we compared the spatial expression of some SOX genes from our test condition, which is the RNase-H, and the lysis, and we also compared that with data from data from the Allen brain atlas for the developing mouse brain, and also from stereoscope. So stereoscope is another spatial transcriptomic approach, and it's really high resolution. So we can, so SOX11 is transcription, I believe it's a transcription factor, and it regulates progenitor and stem cell behavior. And it also acts with other SOX family member genes. And the spatial pattern from using the RNase-H is pretty consistent with what we see from lysis, and is also pretty consistent with the spatial pattern from the stereoscope data for SOX11, and also from the Allen brain atlas image from for SOX11. So we were pretty confident we were able to recapture the expression of some of these genes. We also looked at SOX2, and the spatial pattern from our RNase-H condition is pretty similar with lysis as well as from stereoscope and the Allen brain atlas developing mouse brain ish images. So we decided to proceed with further experiment. So this is a E11 mouse embryo sample. This is an FFPE sample. FFPEs tend to have a more defined spatial structure, which is very well preserved. So on the left is the image of the tissue before the barcoding process, and on the right is the tissue image after barcoding and after collecting the cDNA. What is interesting here is that from on the image on the right, we can see the imprint of the microfluidic channels on the tissue, and this is very important for us because the aim is to combine or co-register this image with the image we'll get from the codex, from the multiplex immunofluorescence data from the PhenoCycler after that. So this image is from this same exact section. So after our workflow, we store the slide in -80. It still works pretty well for codex for the PhenoCycler workflow. So we can see very distinct spatial structure in this mouse embryo. For example, we can see expression of smooth muscle actin here. We see E-cadherin, which is an epithelial marker, really expressed really nicely. So this panel had, I believe, 20 to 25 markers. So I'm just showing a couple representative markers, and we found that we can obtain both really good transcriptome data and also really good multiplex immunofluorescence data from the same tissue section. So in this slide, I'm just showing, comparing the spatial pattern of some genes from the spatial transcriptome and from the codex. So for example, for neurofilaments, we see pretty similar expression patterns, also similar for Ki67. And from this, from the spatial transcriptome, we found an average of around 1,500 genes per pixel, and I believe around 3,000 UMIs per pixel as well. And the size of each pixel is 50 microns. So it would have more than one cell in each pixel. So it's not at for the spatial transcriptome data, it's not at a single cell resolution, but this is what we can leverage the single cell resolution of the PhenoCycler to deconvolve the pixels from our transcriptome data to identify the different cell types within those pixels. So this part of the work, we've been doing it in collaboration with collaborators at Penn and at Yale. So we first start out for the PhenoCycler data. We first start out with cell segmentation. So this cell segmentation, we use DeepCell. It's one of the cell segmentation tools. So the cells are segmented using DAPI and a membrane channel, and then the signals from each pixel are extracted. So all the markers in the signals for each of the markers from the panel are extracted, and then we perform quality control. And the main algorithm that is used here is called MaxFuse. So MaxFuse was published earlier this year, and it's a method that allows you to combine data from different modalities. So you can combine ATAC-seq data with RNA-seq data, or even multiplex immunofluorescence data with single-cell RNA-seq data as well. So we used a single-cell RNA-seq reference from the human, this is from the MOCA database. So it's a database for mouse development. So the single-cell reference was combined with the codex data to annotate the cell types within the codex data. And next, we went ahead to align the image from the PhenoCycler with the image from our spatial transcriptomics. And we did this alignment basically using the DAPI channel. And then subsequently, we do cell to spot mapping. So we map the cells from the PhenoCycler image to the spots in our spatial transcriptome, and then we can perform cell type deconvolution, leveraging the cell type annotated from the PhenoCycler data. And then we can also split the spots into the different component cell types from our DBiT or our spatial transcriptome data, and then we can combine the transcriptome and the proteome data using Seurat weighted nearest neighbor analysis. So on the left is our workflow for the image alignment. So I believe the image alignment was done using an affine transformation, and it was primarily aligned using tissue boundary. So the tissue boundary of the image from the PhenoCycler, and then the image from our DBiT workflow. And then after the alignment and all the other steps I mentioned earlier, we can deconvolve the pixels in our spatial transcriptome data, because our spatial transcriptome data is not at a single cell resolution. So we can't, for each pixel, you would have many different cell types, but by leveraging the cell type annotation from the PhenoCycler, and also knowing the number of cells in each pixels after the alignment, we can assign identities to each of those pixels from our spatial transcriptome data. So for example, this is just showing a zoomed in image of the head section of the mouse embryo. And for example, you can see many different cell types within each pixel. And what is interesting here is that by leveraging the codex or the PhenoCycler data, we can better deconvolve the spatial transcriptome data. So we can pick out features or cell types that would have otherwise been missing if we didn't leverage the codex data. So we also applied this to a healthy human lymph node sample, also FFPE. And this black square here represents the ROI we looked at. So this was from I believe a 25-year-old patient. It's a pretty nice lymph node. It retains the general structures of a lymph node as you'd expect. And we found an average of 1,500 genes per pixel and 2,300 UMIs per pixel. And on the right is a spatial UMAP. This clustering was done using Seurat, and it does a pretty good job of recapturing or recapitulating the spatial structure we see in the lymph node. For example, this section here corresponds to this section of the lymph node. I believe that is where a lot of the vasculature passes through in the lymph node. And in the multiplex immunofluorescence data I'll show, you also see the spatial distribution of B and T cells, for example. And this red cluster is primarily B cells as well. So this is the PhenoCycler image from that same section, and you can even see these horizontal lines, which are imprints of the channels during our DBiT workflow. So I'm just showing a couple select markers, CD20 for B cells, CD3E for T cells, SMA, and CD141, which marks for many different cell types. But it just has a really nice image, so I decided to include that. And we can see that the protein data is pretty consistent with the transcriptome data. We also see the expression of CD20 in a pretty similar region we saw in the transcriptome. Oopsie, sorry. Yeah, so as I mentioned earlier, for example, in cluster 0 here, it expresses genes that are expressed mostly by B cells. For example, MS4A1, which is a B cell marker, which is CD20, and CXCL13, which is also expressed by TFH cells, which are primarily found within the lymphoid follicles. So the codex data or the PhenoCycler data is pretty consistent with the spatial transcriptome data. So this experiment we used two serial sections. One was using our 25 micron microfluidic device, and one was using our 50 micron microfluidic device. I'll just show most of the images from the 50 micron in the interest of time. So we, as I mentioned earlier, we perform our cell segmentation using the Mesmer model from DeepCell. It uses a nuclear membrane and nuclear channel, which is DAPI, and a membrane channel. We use CD45 because the lymph node is primarily immune cells, and most of them express CD45. So we segmented the cells, and this is just showing the expression of some markers, for example, CD20, CD3E, and CD14 in the 50 micron and the 25 micron device. So we also use the MaxFuse algorithm, which primarily or essentially matches single-cell RNA-seq data and codex data, or data from different modalities, and we can see that they align pretty well. So the RNA and the protein data align quite well, and we can also do perform cell type annotation, so we can see the different B cells and T cells. For example, we see a lot of memory B cells in this region. We also see CD4 positive T cells and macrophages and other cell types you expect to see within the lymph node. Oops. So this plot is just a cumulative plot of the different B cell subtypes, which is pretty consistent with expectations for where you would want to see them. And on the right is showing T cell subtypes, and the bottom is showing all the other subtypes, cell types. And this is from the F1 score. We can see that the predicted cell types from the label transfer from the RNA-seq data to the PhenoCycler data is pretty high, 87% F1 scores for the 50 micron and 90 for the 25 micron. So similarly, we also performed image alignment. So this is the image from the codex, from the PhenoCycler data, and is only a small ROI we profiled for our transcriptome. So this is the ROI from the transcriptome data, and those two images are aligned. And following the alignment, we can also do that DBiT spot deconvolution, and we can identify the different cell types that are found within each pixel. And what is interesting here is that from the cell types we find in DBiT, without leveraging the codex data, we are not able to pick out some cell types, or we can't see some cell types don't cluster as well. So using the codex as a pivot or as leverage, we can we have a better ARI and ASW scores, and we can better cluster these different cell types. And similarly, from the using the codex informed deconvolution of the DBiT spot, we can better identify these different cell types. And finally, what is interesting here is that this is basically showing us how much the codex, the PhenoCycler data helps us to identify these cell types, which we wouldn't have been able to do just from the transcriptome, because some transcripts may not be expressed. So it weighs pretty high. It's not for every single cell type though. And what is important here to note is that the ability, our ability to leverage the PhenoCycler data to identify these different cell types in the transcriptome is dependent on the markers we have in our panel. So for example, if our markers don't have a lot of maybe a B cell or T cell marker, we might not be able to tell them apart finely enough. But the biggest takeaway here is that we're able to combine our transcriptome workflow and our PhenoCycler workflow on the same tissue slide. And then by leveraging these two different modalities, we have a better understanding of the underlying biology that is occurring. And finally, this is just UMAPs of some select markers. On the top is the protein markers, and the bottom is the RNA. So for example, for CD20, see pretty consistent expression in these UMAPs. It's not very high for CD4 though, but since we are able to leverage the proteome data, we can get a better picture of the transcriptome as well. So I think that's the end of my talk. I'd like to thank my lab and all our collaborators, and thank you for this opportunity as well. And I'm happy to take any questions.
That's amazing. I think that was a great talk. Are there any questions from the audience? Do we have any questions online?
Nothing.
Okay. Oh, there. Excellent. Yeah, don't worry about it.
Oh, okay. So thank you for the brilliant talk. I wanted to ask, since you did display results with MaxFuse, if you had the chance to check other annotation tools like Glue or Mario, Stellar, and if you had luck with those when annotating your, when not integrating, but basically using them for annotation.
Okay, we primarily use MaxFuse because our collaborators actually developed MaxFuse and Stellar and Mario, but MaxFuse is specifically designed to be able to integrate data sets that are weakly linked. So for example, RNA-seq data and codex data. So we didn't try the others, but I think based on how those algorithms are designed, MaxFuse should in theory give us the best, but it will be worth checking.
Hi, great work. So you're basically using the codex data for deconvoluting your DBiT-seq data. So you touched on it in the end. How do you decide what the optimal panel size and markers are for that purpose? I'm sure it depends on what tissue you're profiling, but you've seen the correlations between the RNA-seq and the protein data, and you also showed that some of them correlate well, whereas for some of them you need the protein markers are more important, where the RNA-seq data lets you down. So would you skew your panel more towards those type of markers, or would you stick with standard markers? I'm sure you don't need CD20 for deconvoluting your RNA-seq. So would you replace that with something which has a lower correlation between RNA and protein?
Yeah. So first of all, this is primarily like exploratory. So if we also study different, we do look at different tissues in our lab as well. For example, we look at DLBCL, AITL, a bunch of other tissues. So in that case, we could consider doing that, replacing some markers which might not be as relevant. But we have a very extensive panel. I didn't mention that, but we typically have close to 45 markers in our codex, PhenoCycler workflows.
That is awesome. So So you are able to get 45 markers off of a PhenoCycler workflow after you have done the DBiT-seq, basically.
Yeah, and most of the markers look really good, actually. So for the lymph node I showed, we also have the PhenoCycler data from a section without DBiT, just as a control, and it's very consistent.
That's awesome. Yeah, so that kind of like goes to show like how we are taking off the fluorophores is not going to affect the tissue much, right? So that's awesome. Thank you. Any other questions?